I am Nikolaos Mellios. I'm an associate professor at the Department of Neurosciences at the University of New Mexico School of Medicine. As you can tell from my accent, I grew up in Greece. I started my career as a physician. I finished the medical school at the University of Athens School of Medicine. And during medical school training, I was immediately excited about the field of neurosciences and the potential for novel discoveries that could be translated for better diagnosis and treatment of neurological and psychiatric disorders. And around in 2012, there were a number of influential papers suggesting that a novel type of non-coding RNAs called circRNAs, which are covalently joined and have a very high stability and also enriched in the brain, there was a number of studies identifying their presence and most importantly their potential functional significance. So this is when I got very interested in circRNAs. I realized the potential of having a molecule that is very much enriched in the brain, and it can potentially be involved in the regulation of brain development, maturation, aging, and of course that increases the possibility that such molecules that will also be involved in neurological, psychiatric, and neurodevelopmental disorders. For the last 7 years, I have been faculty at the Department of Neuroscience at UNM, where I set up an independent lab focused on discovering which circRNAs could be altered in various psychiatric and neurological disorders such as bipolar disorder, schizophrenia, Alzheimer's disease, fetal alcohol syndrome, autism spectrum disorders, etc. So my lab very quickly made a lot of headway on identifying a number of these brain-enriched circRNAs such as circHOMER1 that is involved in psychiatric disorders and is able to robustly regulate brain plasticity and experience-dependent plasticity and learning. So together with a lot of hard-working people in the lab, we were able to find the proper methods to accurately and specifically detect circRNAs and also to functionally test what is their role in different cellular systems and of course the implication for various disorders. One of the biggest challenges in the field of circRNAs is that although there is a very large number of circRNAs that have been identified, close to 100,000 different circRNAs have been detected in the human brain, for example, circRNAs, because of their peculiar shape and because they come within, they utilize the exons and introns of protein coding genes, they are technically difficult to identify, and also the screening for identifying which circRNA could be altered in a given disorder or in a given experimental setting, all of the screenings that we have available are semi-quantitative. So there is challenges on identifying which circRNA is altered, and then there's a bigger challenge on designing proper primers and choosing the proper PCR-based method to accurately and specifically detect circRNAs. We have been utilizing a number of products from Applied Biosystems technologies for both reverse transcription and PCR for detection of circRNAs and other molecules, RNAs, messenger RNAs, and long-non-coding RNAs. One particular quality that we are very interested when choosing such buffers is the fact that utilizing these different chemicals, buffers, and kits in different settings needs to be reproducible. So that's one of the reasons that we decided to utilize Applied Biosystems technologies, because we are trying to minimize any kind of technical error that could come from poor quality of buffers or inconsistent quality of buffers, kits, and assays. I really love being a neuroscientist and a scientist and an RNA biologist. What is particularly exciting is that when you're working in fields of research that are very novel and cutting edge, is that you have the chance to discover something that no one else has discovered. Of course, at the same time, in addition to this intellectual pursuit of discovery, it's also extremely rewarding when your discoveries are translating into clinical benefits and into potential treatments that can help mankind.